Hello, and thank you so much for having me at the 8th plenary meeting of GA4GH. I'm Alice Popejoy, and the title of my talk today is Information Disparities and Implications for Clinical Genetics. Information is key in science, and a lack of it limits our scope and accuracy of our interpretations and inferences. One example I love is in the 1800s, how archaeologists found the remains of a Viking warrior and military strategist that looked morphologically female, but they assumed that a woman couldn't possibly have been those things, and so they still recorded it as male. It wasn't until 2017 that Swedish geneticists actually did a genetic analysis of the remains and found that it was, in fact, biologically a female Viking warrior. Now, this is just one example of how limited information and biases of a sociopolitical context can really limit our ability to make accurate inferences in science. And genetics is no different. In the 1970s, population geneticist Richard Lewontin had this to say about human genetics. What has changed in the evolution of scientific thought, and is still changing, is our perception of the relative importance and extent of intragroup as opposed to intergroup variation. These changes have been, in part, a reflection of the uncovering of new biological facts, but only in part. They have also reflected general sociopolitical biases derived from human social experience and carried over into scientific realms. Now, part of the difficulty of talking about and working in the space of diversity in genetics is that we are all operating in our own sociopolitical contexts, which sometimes are highly controversial. And there are also no standard definitions for diversity measures. So I'll talk about the three main diversity measures that we talk about in the United States. The first one is race. And I did not come up with these definitions all on my own. I rely heavily on social science researchers who have dedicated their careers to studying these things. But when I talk about race, I mean socio-political mechanism for classifying individuals. It's often tied to status and power, and it reifies an ideology of inequality. Ethnicity, on the other hand, although often used interchangeably with race, means something different. It's a cultural construct, often linked to community, religion, or language, sometimes food. And ancestry is something different entirely. We're talking about the biological inheritance of genetic material that can be traced through the genome using genotype data with some reference relative to some geographic region of the world. This is a Venn diagram I put together to demonstrate how fluid and overlapping and slippery these terms are, and the labels that we use to fill those categories in the United States context. I'm not going to talk about all of them, but I'll point out the one in the middle here, Asian. You might say something like, that person is an Asian scientist. Okay, that's a racial stereotype. It's based on someone's phenotype. You looked at them, you made an assumption about their identity and said something racially about the concept of Asian. You might say, last night I had Asian food. That's a reference to an ethnic food group. So that might be an ethnicity reference. And then Asian ancestry. Well, what does it mean to have Asian ancestry when roughly or more than half the world's population lives in an Asian country, and they're highly genetically diverse within them? So not super meaningful, not incredibly scientific, and all of these terms slip and slide between one another, and we're not really sure what we're talking about most of the time. And indeed, again, back to Richard Lewontin in the 1970s, 50 years ago almost, he told us that there's no genetic basis for racial classification. The majority of variation is within these racial populations and not between them. So he said racial classification is of virtually no genetic or taxonomic significance, so no justification can be offered for its continuance. And yet, at least in the United States, we are still using roughly the same racial categorizations, except we call Amerins American Indian or Alaska Native. We turn that Mongoloid category into Asian. African became Black or African American. We also have Native Hawaiian or other Pacific Islander, white from Caucasian, and other. Those are our racial categories that are determined by the US Office of Management and Budget for the census. We also have an ethnic category called Hispanic, non-Hispanic, which can be understood to be a sort of ethno-linguistic category. Now, why am I talking to you about the census categories in a talk about research and medicine? Well, in the 1990s, 1993 to be exact, the NIH passed a rule that said all NIH-funded researchers needed to report back to the NIH the breakdown of their samples by these racial and ethnic categories. And the intent was good. It was actually intended to increase representation and to track the inclusion of different groups in biomedical research that had traditionally been underrepresented. But the problem is researchers kind of took this and ran with it. They said, okay, well, these are the reporting categories, so these must be the analytic categories that are natural to use. And they've integrated these categorizations and stratified samples by these groupings in their research protocols and in their analyses. In the US, we also have these categories in our electronic health records and on clinical forms and all sorts of other data collected in the healthcare system and other systems in the US. So naturally, the clinical genome resource, ClinGen, took an interest in this. And in 2017, with my boss, Carlos Bustamante, we started the Ancestry and Diversity Working Group of ClinGen to look at this question of the use of race, ethnicity, and ancestry in clinical genetics. Just this summer, we published the results of a survey of 448 clinical genetics professionals and researchers in the American Journal of Human Genetics. And there's a lot of data in there. I encourage you to read it. I'm going to draw from a small amount of those data to talk with you about the research to precision medicine pipeline and how lack of information and how the heterogeneity and the way all of these terms are understood and described and used in clinical genetics plays out and has implications for clinical genetics. So clinical genetics still rooted in basic research and basic science, right? We have the identification of genetic risk factors that leads to determining what genetic tests we conduct in the clinical setting. But there's a problem here. There's a lack of diversity in genomics. So the amount of genetic variation or the type of genetic variation that we're testing for in the clinic is heavily biased towards the research, the populations that have been included in research. Also, results are often reported by population, that they're done specifically in these populations and they're reported as such, sort of reifying our beliefs and understanding that perhaps there are these much larger differences between groups than we really know to be true when you look at the data. Also, there are inconsistent population descriptions. So how does this play out in clinical genetic testing? Well, you heard earlier from Dr. Otimi about the lack of genetics across many different sectors and studies. This is a study I did in 2016 comparing to 2009. And I found that 81% of samples analyzed by genome-wide association studies had been conducted on people of European ancestry. But the majority of this diversity here that's been conducted is in Asian populations, and conducted by Asian investigators. So we really still know not very much about African genomes and mixed ancestry individuals, Hispanic, Latino. Actually, the proportion of Native and Indigenous peoples worldwide decreased between 2009 and 2016. I also want to draw your attention to the vast heterogeneity in the population description seen here. It might be kind of small, but hopefully you can look at the slides later. It's also in the supplement of the Nature paper from 2016. We have vastly different types of population descriptors. We have Black cases, that's a heavily racial grouping. Asian ancestry, again, not very specific. What does that even really mean? And then we have all sorts of very obscure specificity here in our European ancestry categories. I had to look up some of them. Corculan individuals and Carlentino individuals, Amish cases, etc. And then we have, Hispanic cases. Again, does that mean all of the people in your sample spoke Spanish? What population is this that you're really talking about, right? So this is the genetics research that's feeding into the research to precision medicine pipeline. We have really no sense of what these findings actually mean for people who don't fall into those categories. And even within those categories, there's so much ambiguity that it's really hard to make accurate inferences. So then once we conduct our genetic tests, we need to interpret them. So we rely on reference databases to look for evidence that the variants that are being found are pathogenic or benign or likely benign, etc. And in order to start looking at this, we were curious about the type of information that clinical labs have about a patient's race, ethnicity, or ancestry. We actually took screenshots of different clinical lab requisition forms from 10 different US-based labs and found vast heterogeneity in the way these categories are described and also which categories are present on the multiple choice forms. When we looked at non-US labs, however, it was slightly different. The vast majority had ethnicity as the label that was used, and we didn't see race or ancestry anywhere. This is an analysis of roughly 60 US and 60 non-US based labs, and 71% of the clinical lab test requisition forms in the US labs had some question about race, ethnicity, or ancestry, whereas less than half, only 47% of the non-US labs had this question at all. And those that were asking about it in the non-US context, most of them actually had a free text option, right? So they're actually just asking people to fill in the blank versus the United States where we typically have multiple choice and then an other category. So that was interesting. And why is this important? Why do we need to know about a patient's ancestry? Well, it's baked into variant interpretation. The ACMG standards and guidelines for clinical variant interpretation have a few different population level evidence codes. And here I'll hone in on this one, it's PM2. It says that if a variant in your patient is absent from population databases, that is moderate evidence for pathogenicity. Now, this makes good sense if the patient you're seeing comes from an ancestral population that is well represented in the database. The absence of the variant means it's likely rare, or it's rare and therefore likely having something to do with a harmful condition. But if your patient does not have their genomic background well represented in the databases being consulted for this exercise, then how do you know whether the absence of a variant means that it's likely pathogenic or that person's ancestral background just is not well represented in the database and that's why you didn't find it? So how reliable is our evidence base for population data, and do we need to factor in the ancestry of the patient that we're looking at? These are all fairly open questions at the moment. So we were curious to ask our survey participants, our clinical geneticists who are involved in interpretation of genetic variants for patients, about the importance of race, ethnicity, and ancestry. And to our surprise, all three, race, ethnicity, and ancestry, were reported by our survey participants to be at least somewhat important for clinical interpretation. Ancestry being the winner here, with more than 45% of survey respondents saying that this is important or very important, and 25% said it's at least somewhat important. So, okay, if ancestry is the important one, how are we getting access to this information? That's pretty sophisticated. We asked our survey respondents how often they calculate genetic ancestry from a patient's DNA for the purpose of clinical variant interpretation, and the vast majority said never or rarely. We asked this question in a bunch of different ways to see if they were getting access to ancestry somehow through the labs or through colleagues or whatever. No, for the most part, people do not have access to ancestry information about their patients. So what are they using? We asked them. If information about a patient's race, ethnicity, or ancestry is used in any aspect of your clinical work, from where are these data obtained? 94% of our survey respondents said that race and/or ethnicity is reported directly to them by the patient. 40% said they got this information from a medical record, but we're not really sure about the provenance of that. Where did that come from? Was the patient asked? Were they not? And 18% actually admitted that race or ethnicity that they use are recorded by another care provider, possibly without asking the patient directly. So what does this mean? This means that for clinical genetics and for the purpose of clinical variant interpretation, the vast majority of clinical genetics professionals are using either self-identified by the patient, race or ethnicity, or clinician identified, meaning someone looks at their patient and checks the box. So once we have an interpretation there needs to be some reporting back to the patient, possibly a diagnosis and treatment, but we know that there's uncertainty across populations, and all of the biases feeding into the pipeline thus far are leading to diagnostic limitations, and in many cases treatment as well. So what are some of those? Well, first and foremost, we have a higher rate of variants of uncertain significance in individuals of non-European ancestry. Not surprising, right? We have information disparity. This limits our ability to make inferences about our findings. We've seen false positive diagnosis rates elevated in African Americans compared to European Americans, where the original study that fed into the association either wasn't diverse enough, so they had a false positive or what have you. Also, false negative diagnoses are more likely in individuals from populations that aren't well represented in clinical databases. This is because we're not capturing the genetic heterogeneity of a disease across populations. If we mostly have information about the genetics that's causing disease in European individuals, we're going to miss variants that are contributing to traits in on other ancestral backgrounds. So the entire research to precision medicine pipeline is fraught with a lack of heterogeneity across the board, lack of consistency in standards, lack of diversity, and therefore limitations in our ability to essentially treat patients equally depending on their ancestry. So we were curious about whether our clinical genetics professionals were satisfied with what they knew and what they were able to do. But the vast majority said, well, 65% said that they believed new guidelines would be helpful to guide the collection, use, and/or communication of race, ethnicity, and ancestry information in clinical genetics. So for the Ancestry and Diversity Working Group of ClinGen, we take this to mean that we have more work to do in working towards these recommendations. And in summary, there is no genetic basis for racial categorization of humans. Hopefully, most of you already knew that. There's also no global standard for the definition, collection, or use of diversity measures in genetics and clinical care. And biases in the research to precision medicine pipeline lead to disparities in clinical genetics. So guidance is needed on the use of diversity measures in science and in medicine, and we're working towards that. So what are our next steps? Well, first and foremost, we need to increase diversity in genomics research and in reference databases. So that means new genomics research that's being conducted needs to be more inclusive of diverse populations, but also the data that we already have on diverse populations needs to be integrated into the reference databases that are most often used in precision medicine and clinical genetics. We also are looking at quantifying this uncertainty in clinical genetics on the basis of representation in reference databases. For example, how do we evaluate whether a database is actually representative of the global population or of an individual patient's ancestral background? Should the patient's race, ethnicity, and/or ancestry actually matter? We're also working to develop a flexible and inclusive framework for human populations to be used in genomics research and clinical genetics. And we really want this to be a collaborative effort. It already is across multiple consortia in the United States, but we would love to expand this to GA4GH. I understand that H3 Africa has actually developed a fabulous ontology. I had the privilege to speak with the phenotypes working group of GA4GH and learned about this really extensive ontology of African populations, which is so important. And we'd love to integrate that and all sorts of other frameworks that folks have come up with so that we can really make sure that this is something everyone can get on board with in our research and in medicine. So we want to engage stakeholders across multiple disciplines and continents to develop guidelines for diversity measures in clinical genetics that will involve both a framework and also data collection and guidance on use for interpretation and reporting test results as well. So I'm happy to take questions, and I'd like to thank Carlos Bustamante, who's my PI and the current chair of the Ancestry and Diversity Working Group, Kelly Ormond, who was a former co-chair, and Sharon Plon, who's our other PI on the Stanford Baylor grant of ClinGen, our program officers for ClinGen and Caesar, Aaron Ramos, Lucia Hindorf, and all of the other folks that made the survey possible that I talked about today. And I'd be remiss not to mention that there are many scholars and investigators whose prior work on the use of race in medicine and diversity in genetics have really made the work that we do possible. So I want to just thank those of you who's the giants on whose shoulders I stand today. So thanks, and looking forward to our discussion.